All right, so this short presentation is going to be on the respiratory system and some of the lab materials that are relevant for the respiratory system. So looking at this picture right here, this is a picture of a model we have, and I want you to focus on just the respiratory structure. So you have the lung, the right lung, the left lung would be over here. This is your trachea coming down, and the trachea is going to branch into a right and left primary bronchus, or bronchi is plural. The primary bronchi enter the lungs, and then they branch. So then we get secondary bronchi, and then eventually tertiary bronchi. And it just keeps branching and branching until you get to the bronchioles. And we have respiratory bronchioles, and then the alveolar sacs at the very ends. Here's a picture of models of the lung. So this is your right lung here, left lung here. The way you can tell the difference is the number of lobes. And the right lung is a little bit bigger, has 3 lobes, left lung, 2 lobes, and it also has the cardiac notch and cardiac impression where the heart fits in. Some more models here. This is a good one that shows the different bronchi. So you get the trachea, primary bronchi, secondary bronchi, and then tertiary bronchi. And here, you've seen this model before, the alveolar sacs, and so you can see that this is a pulmonary artery bringing deoxygenated blood into the alveoli where it can pick up oxygen, and now it turns red. The blood's going to be more red in this model, and this is a pulmonary vein heading back to the heart after the blood has been oxygenated. And this is just showing you the alveoli with the pulmonary capillaries surrounding them. This is a good model of the larynx, and so everything's labeled here, but some of the key points I want to cover. So this is the front, so you get your big thyroid cartilage right here with the Adam's apple, and the laryngeal prominence Adam's apple. Cricoid cartilage is this ring right here, and you can tell this is the back view of the cricoid cartilage. So front, just a narrow little strip. In the back it gets really big, but it's like a ring that goes all the way around. Here's your tracheal cartilage of the trachea and the little annular ligaments in between. Here's your hyoid bone that you remember from the bone chapters. And this is your epiglottis, made of elastic cartilage, and that is going to prevent food, hopefully, from entering the larynx. And when we look at the back view of the larynx, you get these cartilages right here, the arytenoid and the corniculate cartilage at the tips. See, the arytenoid are actually attached to your vocal cords. If you look closely, you can see some little wires right here. These little wires represent the vocal cords. Remember, there's 2 pairs. You have true vocal cords and then false vocal cords above them. And these arytenoid cartilages kind of rotate back and forth, moving the vocal cords, allowing you to speak. And here's a close-up view of that. So you get your cricoid cartilage, arytenoid, and on the tips, the corniculate cartilage. So microscope slide-wise, this is just healthy lung tissue. You've seen this before. So simple squamous epithelium. And this is what it looks like. This is the same slide that you saw back when we did the tissue drawings. And then this is just a close-up view. So you can see it's simple squamous epithelium. These little sacs are the alveoli within the lungs. So looking at a few pathological slides, so normal alveoli, and then emphysema is a condition where the alveoli are destroyed. So you can see, like, this is a good slide of it. You can see the alveoli are destroyed. You get these big gaps in alveoli. Obviously, this is going to hinder gas exchange in the lungs, and so people with emphysema have trouble oxygenating the blood because they lose all the surface area of the alveoli. Here's another picture where you can see some gaps in here. So both of these are emphysema slides along with this one, and identifiable because of these big gaps in the alveoli. This is a microscope slide of pneumonia, and in pneumonia, commonly it's due to bacteria getting into the lungs, and the alveoli fill with pus and fluid. So you can see these little, and even microorganisms you can see inside the alveoli. So identifiable by the alveoli filling with pus and fluid, and you can also see tiny microorganisms in there. So commonly caused by bacteria. So that is pneumonia. And this is a slide of lung cancer. And so lung cancer, the cells replicate very quickly. And so what you see in the alveoli in lung cancer is just tons of cells. So all these little cells replicate really quickly. So it's just like tons of cells in there, lots of cells. And that's how you would identify a microscope slide of lung cancer. Or a couple other slides for you. This is a cross section of the trachea and the esophagus. So the esophagus we obviously will talk more about with the digestive system. But this is the trachea, so it's nice and wide open. The esophagus is closed, but so what keeps the trachea open is all the cartilage, the tracheal cartilage. And if we zoom in on the trachea, you can see it's lined with pseudostratified ciliated epithelium, and you can see that closely right here. So pseudostratified ciliated epithelium. There's goblet cells in there that secrete some mucus, and then here's the actual cartilage of the tracheal rings. So it's hyaline cartilage. So trachea, esophagus.